Good morning. We have today Regeneration scientists, everybody else. I'm a scientist here. So I have three main research areas in my lab. So my lab overall works on how certain animals can regenerate, but humans have very limited capacity to regenerate. So we're very interested in scar regeneration, spinal cord regeneration. And for this, we use the Eldestone mexicanum, commonly referred to as an axolotl. It's a type of salamander. And then we also use a local species of sea anemone called Nematostella victensis. So I'm going to tell you a little bit about both of these areas, and then I'll tell you about the course, which is related to research. So this is our axolotl. We call it the champion of regeneration. So it can regenerate multiple body parts, including the forelimbs, the hind limbs, a portion of the heart. You can make lesions in the brain. You can make a lesion in the spinal cord, and it all fully regenerates, and it is functional regeneration. So why do we use axolotls? Obviously, it's the champion of regeneration, but it also has very big embryos, as we're looking at here. So it takes a month to develop into what is a tiny little axolotl. We can raise a large number of these animals in the lab. The genome is sequenced, which allows us to do genome editing. We can make transgenic lines relatively easily. A transgenic line means we can put in a fluorescent protein to allow us to visualize certain types of cells. In panel A here, we're looking at a transgenic line, which allows us to visualize all of the neurons in the animal. And in the other panel, we're looking at transgenic line, which allows us to visual the cartilage, which is very important for when we're looking at limb regeneration or vertebrate regeneration. In vivo gene modulation is possible. So you may have heard of this technique called CRISPR-Cas9, which is simply a technique which allows us to knock out a gene or insert a new piece of DNA into the genome. So this is an example of limb regeneration. So we can amputate here at the level of the hand or up here at the level of the forearm. And what you see is over time, cells crawl over and heal over the wound. And then we have this very specialized structure between the blastine, which is basically it's a mound of proliferating cells, which is eventually going to replace, in this case, the hand, or in this case, the lower arm and the hand. So somehow these cells at the plane of amputation actually know exactly what to regenerate. And this is the case where you're regenerating a limb or a part or a portion of the brain. We focus a lot on how these animals will actually regenerate a functional spinal cord. So this is just showing you an example in humans. If we have injury to the spinal cord here, we form what's called a glial scar. So there's a scar tissue, which is good and bad. The good side of it is it helps to stabilize the injury site and prevents more injury occurring. The bad side is that it expresses a lot of proteins which are inhibitory to axons growing down, which means that we cannot regain function. In comparison, it's been described many years ago that if you look in a salamander, it appears that if they have an injury to a spinal cord, both sides migrate towards one another, and this allows the axons to grow through your injury site, and the animal regains function again. So this is a large portion of what we study in the lab. And so the axolotl is a vertebrate, but the other animal that we're actually interested in, that we work with in the lab, is a sea anemone, as is shown here. And these are native to the shores here, so you can find them up in Silver Marsh. They're found all along the east coast of the US. And so why do we use these? It's because it gives us another more simple model to study regeneration in. And development is very fast. So I said the development in the axolotl was fast, but it's all relative. So it takes about a month to have a tiny little axolotl. In this case, you go from a single egg to what resembles a very small Nematostella within a week. So this is, it's very optically transparent as well. The genome again is sequenced, and we can make transgenic lines, or we can visualize certain cell types. So this is just showing you that whether you look in these young animals here or in adult animals, so what we're looking at is it's pharynx, and it's got these tentacles. So just like a any type of jellyfish, you can amputate actually any place in the body. And what you're looking at here is tentacle regeneration. So within about 10 days, it will fully regenerate. So As I said, these are found up in the Great Wizard Marsh. And so another area of research which we use these animals for is actually to collaborate with our colleagues in ecosystems in the Bay Paul Center. And we know that over years, the numbers in the marshes have been decreasing. And part of the reason is that more and more people are living in the areas around the marshes, so there's more pollution. So one of the studies we've done is looking at how environmental pollutants leads to defects in growth. And what we're looking at here is a control animal. And these are animals. Phthalates is a component of plasticizer, so this is considered a microplastic. And here we're looking at potassium nitrate, which is commonly used in fertilizers, which are used for the lawn. And so we can incubate the early embryo in these microplastics or potassium nitrate, and then look at how it affects the development. And what we found is overall the body size is much smaller and that they're actually failing to develop tentacles. And so how is this related to the course that I teach? So I'm teaching here a course in stem cells and regeneration, going from aquatic organisms to animals. And so this is currently part of the UChicago Spring Quarter. So this has been the third year that I've taught this course. It's a 3.5-week intensive lab course. So the objectives are that the students will learn much more about animals which have the natural ability to regenerate. They gain hands-on experience with a large number of animals. And this down here are examples of the animals we use this year. This is the zebrafish. We use actually 3 different types of cephalopods, which are bred here at the MBL. We use these Nematostella. We also use flatworms called planarians, which regenerate using stem cells. And then we also use the axolotl, which is the main organism in my lab. And so in the first week of the course, the students get to work with all of these animals, and then they eventually design and carry out a mini project on regeneration. And the way this course is structured is that in the morning, they have a lecture from 9 to 9:50 they should read. And then after that, we do a journal club where students discuss primary literature. And then in the afternoon, it's lab work. And so we have students of all levels. They don't need to have a background in developmental biology to take this course. Actually, this year we had a lot of environmental students. And so our work on Nematostella and the blue that's very popular for projects for the students who have an environmental background. With the journal clubs, we pair students of different levels, and some who have a background in developmental biology, some who don't. So they discuss the papers together in groups. And then this is just some pictures of what students are physically doing in the lab from the course this year. So they learn how to microinject, they do CRISPR-Cas9 genome editing in zebrafish embryos, and they do a lot of imaging as well. So I'm going to leave it there.
Thank you.